All right, everyone, it's time for the functional physiology introduction to cardiac physiology. And to understand that, let's just go over some brief things about the electrical conduction system. So one thing that's cool about the heart is that it runs itself, essentially, and it does that by receiving nervous input. Whoa, come back, buddy. Receiving nervous input that connects to the node that sets the resting heart rate, and that's called the sinoatrial node. So there would be a nerve that would come here, maybe release acetylcholine. Yeah, definitely acetylcholine. Acetylcholine would bind to its receptors, which are muscarinic in the tissue, and excite this SA node. So the SA node and the next node, the AV node, the atrioventricular node, they are made out of a tissue that's more similar to the endocardial tissue. So these are essentially like nervous pathways that overlay, or specifically underlay, the myocardial cells, so the muscle cells. So what they do is conduct a charge which can then spread through the myocardial cells to stimulate them to contract. So we have first this excitation happening at the SA node. So we call a normal heart rate a sinus rhythm because the sinoatrial node should control resting heart rate. And once that hits there, we'll see this wave of contraction spreading through the atria, and then also spreading down to the next node, the AV node, which can then be excited, and then spread that wave of excitation all the way down the bundle of His, the right and left bundle branches, and then up through the Purkinje fibers. So every time the heart beats, we have stimulation here, spreading from the SA node to the AV node, down the bundle, through the bundle branches, then up through the Purkinje fibers. So this is the conduction system. If you know the pieces, that is one step that you need to understand, or in understanding the way that the heart works. And these are types of cells you've probably learned about before called excitable cells. Excitable cells can generate action potentials. They're not always on, they only turn on when they are told to do so. And action potentials, an action means whatever that cell's job is to do. So it's a heart muscle cell, its job is to contract. So we can even call them contraction potentials. And what potential means is charge. So what is the charge that you need to get to have a muscle contract? That is what an action potential is, getting to that level of excitation for contractions. And action potentials, they're kind of like plugging something in. They're supplying power to it so that it can work. So here we have the SA node. It's receiving this stimulation. And it can then spread, because remember, all these things are just talking in ions, like positive ions, spread that current through ions down through the myocardial cells, which are connected to one another. So see these fun little things, those are intercalated discs. Those are specialized gap junctions, there we go, gap junctions, that allow the free exchange of ions from one cell to another. So we don't have to wait for any stimulation, we can just spread those positive ions and they can go channeling on down one cell at a time, spreading this wave of excitation that allows those cells to contract. So here is what we see inside of these cells. We have these gap junctions, so free exchange of ions. We have T-tubules, which you've also seen in skeletal muscle. Those allow the spread of ions down into these junctions. We have a specialized endoplasmic reticulum, a smooth endoplasmic reticulum called the sarcoplasmic reticulum, and that stores calcium. And calcium is a positive ion. When calcium is liberated from the sarcoplasmic reticulum, it makes the cell more positive, it depolarizes it, and it increases the chances that there will be an action potential. Here's a little intercalated disc. It's a fun word to say. Say it to yourself. Intercalated. Wasn't that nice? And inside we see our favorite little myofibrils. We've got our actin and our myosin and the sliding filament of contraction means that these filaments will slide towards one another until this zone disappears, and then they go back to what they looked like before. So you should know that calcium then is very, very important to contraction, and especially contractile strength. The more calcium there is in the extracellular fluid, the stronger the contraction will be of a cardiac muscle cell. Another fun word to say is syncytium. Syncytium. Isn't that fun? Syncytium. Syncytial tissue is tissue that works as a sheet. So this tissue is all connected. When you excite one cell, it spreads that excitation through positive ions all the way down so that this cell isn't working in a vacuum. This tissue works together as a sheet. Syncytial, rapid spread of excitation. Look at that. One direction, syncytial tissues working together as a sheet. Okay, there's two syncytial tissues. There's the atrial syncytium and the ventricular syncytium. When we have stimulation of the SA node, that syncytium is excited so that we have atrial contraction. Eventually, that wave of excitation makes its way to the AV node, which will then spread down through the ventricles. This is important, believe it or not, because when there is damage to these areas, that causes abnormal heart rhythms, like the Winky Bach, which is a really fun song also. So you can see normal atrial contractions, for example, but you can have abnormal QRS complexes if there's physical damage to the bundle or to the AV node. All right, so this is what the action potentials look like in ventricular muscle and Purkinje fibers. I want you to notice the, whoa, look at all these things coming up out of nowhere. This is the fast sodium channels. We'll talk about different types of tissues in the body. The muscle, so those are our myocardial cells, and this is the nervous tissue, the Purkinje fibers. So you can see that when the Purkinje fibers are excited, we will see contraction of the ventricular muscles right around the same time. So action potentials, they prime the muscle to contract, so they prepare the cell for contraction. The heart contains fast sodium channels and slow sodium channels. There's a decrease in calcium permeability that leads to slow recovery. One other thing that we've mentioned about other action potentials too is there is a time when there should not be another stimulation. That is called the refractory period. So the refractory period is when it is physically impossible to have another action potential. So technically, when this action potential is happening, we call that the absolute refractory period because the cell's already at that charge, so it can't be at that charge a second time. That doesn't make sense. Relative refractory means it's really difficult to have a contraction during that time, but not physically impossible. It can happen if there's too strong of a stimulation to the system. But think about what's happening in the heart. When there's contraction, the ventricles are ejecting the blood, and then for that, like half a second right here, the heart needs to refill again. So every time we have contraction, we should have this nice filling period. If not, this stimulation can lead to a premature contraction. So that is going to lead to not enough blood being ejected and then an increase in heart rate, and that feels a little weird when it happens. It's like the heart will have to skip a beat to get back to systemic pressure. There's later premature contractions, which don't really happen during the relative period. It's just still way too soon. The less time for filling you have, the lower the stroke volume. The lower the stroke volume, the more the heart rate will have to increase to sustain systemic pressure. Okay, another thing that you might remember from muscle is that not only is calcium important, calcium is stored inside of the cells themselves. As I've mentioned, the specialized ER, the sarcoplasmic reticulum, that is what stores our stored calcium. I want that to go away now, but calcium. When there is, as I've mentioned, calcium in the extracellular fluid, the calcium will come down here through the tubule, enter the actual cardiac muscle cell, and then bind to its receptors here. They're called ryanodine receptors, and actually cause the ejection of more calcium. It's called calcium-induced calcium release. And there's so much calcium that can come out of there. It causes this nice big contraction called a calcium spark. I'll put up a quick thing to a video to show you how cool it is when these calcium sparks occur. You can actually track the calcium going through a myocardial cell and causing contraction. Eventually that calcium will be pumped on up back into the sarcoplasmic reticulum, just using a standard ATPase to bring the calcium back in and then store it for the next time we need it. There's also, like in every cell, sodium-potassium pumps to maintain the charge, and in cardiac muscle cells, there's sodium-calcium antiporters as well to help maintain the charge. So these pumps, as long as there's ATP in the body, will maintain calcium entry and keep calcium stores stable so that they're always available for muscle contraction. All right,